Friends, till now we have seen the basic concept and terminologies related to bacterial taxonomy and the three interrelated parts of taxonomy, that is classification, nomenclature and identification. So to move further, now we will focus on taxonomy and important feature of some important probiotics like lactobacillus and bifidobacterium. So first let's have a look on taxonomy of lactobacillus. The genus Lactobacillus is taxonomically complex and it is composed of over 170 species that cannot be easily differentiated phenotypically and often requires molecular identification. Although they are the part of normal human gastrointestinal and vaginal flora, they can also be occasional human pathogens. They are extensively used in variety of commercial products including probiotics. According to taxonomic outline of probiotics, the genus Lactobacillus belongs to the phylum Firmicutes, class Bacilli, order Lactobacillus, family Lactobacilliaceae. Now we will look at taxonomy and classification. The closest relatives of Lactobacillus being grouped within the same family are the genera Paralactobacillus and Pediococcus. The phylogenetically closest family appears to be Leuconostocaceae family, which includes genera Leuconostoc, Enococcus and Vizela. In the earlier approach, Orla Jensen subdivided Lactobacillus into the genera Beta-Bacterium, Thermobacterium, Streptobacterium, Streptococcus, Beta-Coccus, Tetracoccus and Microbacterium on the basis of their morphologic and phenotypic features. Today, only the name Streptococcus is still valid, whereas Enterococcus, Lactococcus and Vagococcus have been separated from the original genus, that is Streptococcus. Taxonomy of other lactic acid bacteria genera has also undergone considerable changes since the time of Orla Jensen, resulting in the genera which is listed in the following table. Such taxonomic knowledge of a strain may therefore give an indication of strain's origin, habitat and physiology, and have more important consequences for the selection of novel strains for application in food fermentation or for use as probiotic. According to metabolism, Lactobacillus species can be divided into three major groups. First one is obligatory homofermentative, that is group 1. It includes Lactobacillus acidophilus, Lactobacillus delbrueckii, Lactobacillus helveticus and Lactobacillus salivarius. The group 2 includes facultatively heterofermentative group. It includes different species of Lactobacillus like KZ and many more. The third group is obligative heterofermentative. It includes Lactobacillus brevis and Lactobacillus other species. After classification, let's have a look on important features of Lactobacillus. Here, basic morphological, biochemical, cultural and physiological features are considered. In morphological features, the first one is gram nature. By studying the gram staining, we can come to know whether the organism is gram positive or gram negative, which is based on their cell wall composition and the lipid content in the cell wall. As far as Lactobacillus is concerned, it is gram positive and appear violet in color, and it is non-acid fast, that means it takes gram stain readily. The morphology, that is the shape of the lactobacilli, is rod or bacillus having the size of 0.5 to 0.8 micrometers. Next feature is motility. By doing hanging drop method, we can come to know whether the bacteria is motile or not. Motility based on the type and number of flagella. Lactobacillus do not possess any sort of flagella, hence it is non-motile. Next important morphological feature is capsule, as it is responsible for virulence, that means ability to cause a disease. Lactobacillus is non-capsulated, hence it is non-pathogenic. In addition to above morphological features, one important feature is pore forming ability. Lactobacillus is non-spore forming organism. Now let's have a look on biochemical feature of lactobacillus. As far as sugar fermentation is concerned, here production of acid and gas is checked. Among the sugars, lactobacillus ferments only glucose and fructose, whereas it is lactose and maltose non-fermenting. If you look at the type of metabolism, lactobacillus species is obligatory, homo and heterofermentative as well as facultatively heterofermentative. The third characteristic is cultural features. For cultivation of lactobacillus in laboratory, MRS media is generally preferred. Here, lactobacillus gives round, wavy, flat, mucoid, dry, white creamy color colonies. The next criteria is physiological and other miscellaneous features. Apart from morphological, biochemical and cultural features, physiological features are also have significance in cultivation of organisms. Hence, the optimum temperature required for the growth of lactobacillus is 37 degrees Celsius, but it can tolerate the temperature from 25 to 50 degrees Celsius. If you look at the pH condition, the optimum pH varied from 5.5 to 7, at which lactic acid bacteria gives maximum growth. As we all know, metabolism is an important feature for every living organism, and in metabolism, enzymes play an important role. Likewise, lactobacillus also produced one key enzyme, that is catalase, which can act on hydrogen peroxide and evolve oxygen gas, where it is not having ability to degrade urea as it is urease negative. So friends, in this module we have seen the taxonomy and important features of lactobacillus as one of the probiotics. Here we have focused on taxonomy and classification of lactobacilli. Further, we have discussed morphological, biochemical, physiological and metabolic characters of lactobacillus.